This presentation is realized in the framework of the IUFRO Task Force, strengthening Mediterranean nursery systems for forest reproductive material procurement to adapt to the effects of the climate change. The presentation is focused on the nursery chain and forest reproductive material in the Mediterranean, trying to show how much the nursery system in the Mediterranean is ready to face the challenges deriving from climate changes. Mediterranean forest ecosystems harbor a vast number of flora and fauna species, and for this reason are considered biodiversity hotspots. This important characteristic is challenged by different threats that can cause alteration and failure of the natural regeneration of forest ecosystems. Consequently, artificial regeneration with appropriate forest reproductive material, FRM, is the option to be followed if we do not want to lose precious ecosystem. This means that production, trade, and use of genetically and adaptively appropriate FRM is critical for the future of Mediterranean forests and the goals and/or services produced in the face of climate change. This is particularly true considering the role that restorative activities are receiving to fight climate change, sustain biodiversity, improve resilience, and re-establish an ecologically healthy relationship between nature and culture. At the global level, many initiatives are ongoing, aiming at restoring millions of hectares during the next decade, necessitating many millions of established plants. These initiatives and activities are important in the Mediterranean region, as climate has changed in this region faster than global trends. The projections for the next decades show that warming over land will likely be in the range of about 1 to 5.5 degrees Celsius, according to different scenarios. As regards precipitation, the projections forecast a mean rate of land precipitation decrease around 4 to 22%. The concurrent change in temperature and precipitation will affect the distribution of arid lands, with an extension to higher latitudes of regions facing water limitations. The change in climate will be coupled to a change of the species distribution range, with the species-specific responses to drier conditions that could lead to shifts in plant community composition, favoring the most drought-resistant species. In general terms, species at the rear edge of their distribution in the Mediterranean and mountain areas will be the species most threatened by climate change, whereas the most xeric Mediterranean species, which are better adapted to drought, are those expected to encounter fewer problems for survival and range expansion under future climate change. This can be observed in this simulation for a temperate broadleaf summer green and a temperate broadleaf evergreen that emphasizes the likely risk of irreversible losses of many endemic forest tree species and/or populations or unique marginal peripheral ecotypes with their genetic values. In conclusion, to meet the needs posed to the Mediterranean basin due to climate change for highly adaptive genetic material, the production, trade, and use of forest reproductive material is a critical issue. The categories of forest reproductive material appropriate for transfer and use for agroforestry and forestry have been defined at the international scale and at the European level. At the international scale, they have been defined by the Organization for Economic Cooperation and Development, OECD, through the OECD scheme for the certification of the forest reproductive material moving in international trade, released in its amended form in 2018. At the European level, the categories were defined by the EU Council Directive 1999-105 EC on marketing of FRM. There are 4 categories of FRM intended mainly for forestry and agroforestry. The first one is source identified, which is the minimum standard. The material may be either a seed source or stand located within a single region of provenance. The location and altitude of the places of collection must be recorded, while little or no phenotypic selection has taken place. The second one is selected. That means that they are made by basic material of a stand located within a single region of provenance, which has been phenotypically selected at the population level. The third one is qualified. Thus, basic material shall be seeds, orchards, parents of families, clones, or clonal mixtures. They are phenotypically selected at the individual level, and undertaking or completing testing is not required in this case. The last one is tested. So basic material consists of stands, seed orchards, parents of families, clones or clonal mixtures whose superiority must have been demonstrated by comparative testing or genetic evaluation. During the identification and management of basic material, the stock of genetic diversity should be maintained as high as possible. But the risk of losing genetic diversity can occur at different stages. For example, when collecting seed from only few seed stands, or when collecting from few trees within each stand, or when few clones participate in seed orchards, when panmixia during reproduction is lacking both in seed stands and seed orchards, during extraction, cleaning and sorting, during plant grading and culling in nurseries, during storage when associated with loss of viability, or due to lack of stratification in order to break bud dormancy. Activities taking place in nurseries are usually the most critical in affecting the genetic variation of FRM. For example, when selection of seed lots is based on seed properties, for example, lot price and germinability, differing germination response may cause unintentional unrepresentation in seedling. Pest management and fertilization may alter the genetic composition of seedling lots. Transplantation and undercutting of seedlings are techniques often applied to optimize profitability, but may also have genetically negative selection effects on the seedling lots. Genetic variation can be affected also, for example, by mycorrhization. For mycorrhization, functional compatibility among the symbiont is a prerequisite. Also, grading or culling based on size, for example, seedling height or stem diameter, etc., may influence the genetic composition of the seedling. Conditions and timing of seedling storage may affect genetic variation of a seed lot. And finally, production environment caused by acclimatization of seedling to the nursery optimal conditions can have potential epigenetic effects. To overtake the limits that can arise from using not inadequate basic material, the need of certification procedures that make available to the forester seed and plants that are true to name and satisfy certain minimum requirements of quality is a clear consequence. The object of the certification of tree seed and plants is to maintain and make available to the practitioner forester sources of seeds, plants, and other propagation materials of superior provenances and cultivar so grown and distributed as to ensure the genetic identity and high quality of the seed and plants. This was the intention of the already cited EU Council directive, asserting that FRM must be identified throughout the whole production chain to be able to verify that the reproductive material is the same as the declared provenance. This is essential because FRM must be adapted to the planting site, particularly in terms of its stability, resistance to pests and diseases, adaptiveness and adaptability, as well as the productivity and diversity of the future forest. Thus, national authorities should use an electronic database to store all the data about FRM suppliers, basic materials, issued master certificates, field control protocols made by the authority, and supplier documents. This is the model certificate reported by the directive named Master Certificate of Identity for Reproductive Material, reporting data on the identification, the origin, the status, etc. The identification throughout the whole production chain requires the ability to trace the basic material by a unique reference that needs to be provided on the master certificate after each harvest. It is a key element for tracing FRM. The legal control regulations imply the monitoring of the master certificate, but such a monitoring scheme cannot be secure enough. Alternatively, the development and use of genetic markers offers new possibilities for precise and efficient control based on the comparison of the genetic composition of seed stands, seed and seedlings within a traceability system. Trade and transport can also negatively affect the genetic value of the FRM lots planted. With the EU Commission Regulation EC 1598/2002, the record keeping of FRM transfer is needed on a national basis, besides collaboration between national authorities. But harmonization of EU documentation is far to occur. In many cases, forest nurseries and suppliers, for example, sell their products on the European market, and the absence of barriers also allows nursery companies to produce seedlings by subcontracting growers in other member states, which means that seeds and seedlings are transported across the EU during both the marketing and production phases. For FRM lots transported across countries, specific requirements need to be fulfilled during handling, packaging, and storage of the seedlings. Container and bare root seedlings often have differing needs that need to be considered. Seedlings transported in the dormant state are less sensitive, for example, to damages due to transport. Containerized seedlings are less sensitive than bare root seedlings to environmental stress during transport. Seedling storage prior to planting is usually not fulfilled due to lack of cold storage facilities. But what is the current situation for FRM used in the Mediterranean basin? FRM of different genetic diversity and quality may be currently used in different countries, EU and non-EU, across the Mediterranean basin, depending upon the framework regulating its use within each country, Forest owners may purchase FRM and not basic FRM as defined by the OECD and the EU Council Directive. Most of the foresters may not fully aware that the genetic quality of a given basic material may vary across years, depending upon factors affecting parmixia, while extreme deviations can bring to poor adaptation of FRM to afforestation sites. What needs to be done thus? Legislative regulations frame securing the sufficient FRM genetic diversity used for reforestation and afforestation purposes needs to be adopted across all countries of the Mediterranean basin. Methods of collection should secure the preservation of species-specific gene pools and prevent the genetic erosion of seed lots when compared to the source of origin. Forest nurseries should follow specific guidelines in the production line of FRM material that would secure the above goal of existence of sufficient genetic variation in the produced seedlings. Forest owners need to be informed on the significance of using genetically diverse and well-adapted FRM. And finally, seed orchards managers need to focus on methods of securing diversity of the seed crop and on methods assessing the level of diversity in the SO seed crop prior to cone collection. All these needs can be satisfied following the guidelines reported in the Forest, Seed and Plant Scheme by OECD. However, its application is not diffused, as mainly European and North American countries participate in the scheme and 5 African countries outside the Mediterranean area. Thus, it is necessary extending its application to other countries of the Mediterranean region that likely will need to exchange FRM in the near future. The source of FRM for the Mediterranean region was compiled for conifer species more than 20 years ago. This was an effort of FAO Silva Mediterranea and other international networks that showed the need to establish and harmonize shared databases and maps of species distribution. The existing trial networks are precious and should be carefully conserved, managed, and monitored to collect valuable information on adaptation and to integrate marker-assisted selection techniques. It's time to review this directory, integrating the data with other species and information on their health or marginality, as an example. This is one of the main goals of the task force that has produced a platform to collect and store data and metadata of existing sources of FRM in the Mediterranean region, supported by FAO Silva Mediterranea and its working group Forest Genetic Resources. This is a quick overview of the type of required data divided into mandatory and optional. The required fields cover data on identification, legal status, the existence of specific threats, their marginality, etc. To conclude, it is important to harmonize the use and transfer of FRMs across the basin, establishing national strategies that create demand for good quality FRM and sharing good practices to improve FRM production, use, and conservation. It is necessary to keep national and international FRM registers updated by recording not only the origin, but also the movement of FRM for traceability. This would require information system of georeferenced records of the origin, movement, and use of FRM that annotate the plots in which the reproductive material is planted. Monitoring the plantation success would allow to assess the adaptive capacity of plant materials used across a spectrum of sites. Thank you very much for your attention. For requests or further clarification, feel free to contact the coordinator and deputy coordinator of the task force.